lamentamos el atraso en partir, pero ya está todo andando perfecto. Así que para nosotros es un tremendo honor hoy día tener en esta charla como parte de las actividades del Instituto Milenio en Inmunología e Inmunoterapia y también del grupo de microbiología e inmunología celular, molecular y translacional de la Facultad de Ciencias Biológicas. Como speaker al doctor Alessandro Sete. Me gustaría dar la palabra al doctor Calergis para que pueda comentar algunos aspectos del doctor Sete y partamos la presentación. Perdón, ¿puedes presentar acá? Estamos transmitiendo desde el...
Estaba diciendo que durante la pandemia la colaboración científica fue clave y en particular el grupo de Alessandro con Alba y Daniela. Alba ha estado acá 2 veces, so Alba's been here twice, I think. Fue genial porque nos compartieron materiales varios de los que ustedes están acá han utilizado para sus experimentos y aportaron. Así que, em, además de un gran inmunólogo, un gran, digamos, persona, muy generoso y abierto a colaboraciones. Me estaba contando recién una colaboración que tiene, que no sabía, muy potente con personas del Albert Einstein en Nueva York. Así que, bienvenido, welcome. Habla español, así que le pueden hacer habla, pero no tan fluido, pero algo habla, porque es de origen italiano, ¿no? Pero eso, la doctora bueno va a hacer la presentación más curricular.
Muchas gracias, doctor Calergi. Sí, voy a dar un pequeño resumen de su CV. El doctor Alessandro Sete, bueno, es un investigador italiano de origen, pero que actualmente trabaja y por bastantes años, bastantes décadas en Estados Unidos, en California, San Diego, y se ha dedicado más de 40 años a comprender los mecanismos básicos del reconocimiento de antígenos. Y esto, por supuesto, llevando a la respuesta inmunitaria. Medir y predecir la actividad inmunitaria y a desarrollar estrategias de intervención contra el cáncer, las enfermedades infecciosas, las enfermedades autoinmunes y las alergias. Su laboratorio define químicamente las estructuras específicas, que son los epítopos, que reconoce el sistema inmune y utiliza este reconocimiento para medir y comprender las respuestas inmunitarias. En los primeros trabajos que realizó a mediados de los 80 y 90, demostró que la función biológica principal del complejo mayor de histocompatibilidad es la unión de epítopos, lo que es un descubrimiento y una descripción sumamente importante para la inmunología. Y a partir de estos estudios se profundizó en la idea de que los diferentes complejos mayores de histocompatibilidad presentaban especificidades de unión distintas que pueden utilizarse para predecir epítopos, y su grupo, por lo tanto, se ha centrado en identificar estos motivos para que cerca de aproximadamente 100 variantes de moléculas de MHC de clase 1 y 2, tanto expresadas en humanos como en diferentes tipos de especies. Lo interesante de esto es que desde el año 2003, el doctor Sete ha supervisado el diseño y la gestión de una base de datos nacional de epítopos inmunitarios, que es el IEBD, que es un recurso bioinformático gratuito y accesible a todo el mundo y de amplio uso. Esta base de datos, el IEBD, cataloga todos los epítopos humanos, primates no humanos, roedores y otros vertebrados, incluyendo alergenos, enfermedades infecciosas, autoantígenos, trasplantes, incluye herramientas de predicción de epítopos para acelerar la investigación inmunológica en todo el mundo. Y una cosa interesante también es que año a año hacen un curso de entrenamiento también online a nivel donde asistentes de todo el mundo participan, en donde se muestra cómo funciona el uso de esta base de datos y que por supuesto ha tenido un tremendo impacto para el estudio de la respuesta inmune T alrededor del mundo. Entonces, el doctor Zeta actualmente se ha enfocado, bueno, en estudiar múltiples enfermedades infecciosas y incluyendo la respuesta inmune contra VIH, hepatitis B, hepatitis C, enfermedades emergentes y enfermedades con potencial de pandemia y con importancia en biodefensa. Hizo una contribución muy importante para el estudio de la respuesta inmune inducida por la infección natural por el virus SARS-CoV-2 y también las vacunas que se desarrollaron contra este patógeno. Y actualmente también, por supuesto, incluye dengue, zika, chikungunya, malaria, tuberculosis, pertusis y herpes. Entonces, ha adaptado estos métodos y estas técnicas desarrolladas en el contexto de enfermedades infecciosas para justamente comprender la respuesta inmune T ante alergenos que son más comunes y muy recientemente, que es algo que es muy interesante, sobre todo en el área de la interdisciplina entre la neurociencia y la inmunología, de describir el componente celular inmunitario para enfermedades como Parkinson y recientemente la esclerosis lateral amiotrófica. So it's a great pleasure to have you here. So thank you very much for coming to Chile. He also was in Valdivia the last 2 days, also showing his data to the scientific community in Valdivia. So thank you very much, and the floor is yours.
Yeah. Okay, so. Thank you, Susan. Thank you, Alexis, for those nice introductions. So the title of this lecture is The Study of Conserved Viral Regions in the Context of Pandemic Preparedness. And actually, most of the data that I'm showing you is unpublished. But really what we have been doing lately starts from lessons that we learned in the context of SARS-CoV-2 pandemic, particularly in the context of the pandemic and in the early study of the pandemic at the level of infection. This is before the vaccines were available. Clearly, it was pointed out that it's important to understand that is different whether you're considering protection against infection versus protection against disease. And in the context of protection against infection, the most important effector arm of the immune system is the production of neutralizing antibody responses. But when you're talking about protection against severe disease, hospitalization, and death, it is important to consider the fact that T-cell immunity and cellular immunity in general play a much more prominent role. So what is the evidence that shows that that is indeed true and that there is a prominent role for cellular immunity in the context of SARS-CoV-2 immunity? One early evidence was, can you see the cursor? Well, so one early evidence was came from studies where we looked at the immune response early following SARS-CoV-2 infection. And we saw that the best correlate on having a mild disease as opposed to having a severe disease was to have early CD4 T-cell responses and also CD8 responses. And the early antibody response actually did not correlate with disease severity at all. This data was also paralleled by studies. And the first study really was the study from Bertoletti's group in Singapore. That study really showed that having an early, in the first few days after infection, an early T-cell response correlated with having a lower viral titer and a shorter duration of viral positivity, which obviously makes sense if a T-cell response can limit the viral production, that also can translate in a lower disease severity. In another series of studies, what we did was to look at samples from Italy. You might recall that Italy was hit very early and very hard by the SARS-CoV-2 pandemic. And so we looked at samples that we have obtained from a number of collaborating labs. And we saw that a correlate of having a less severe disease was to have a response that was broader. So people that targeted multiple antigens in SARS-CoV-2 beyond spike and also targeted nucleoprotein and non-structural protein had a better disease outcome. The other observation that came out of the SARS-CoV-2 pandemic is that T-cell responses cross-reacted very effectively across different variants. So you, of course, remember that in the SARS-CoV-2, then started to come the alpha, beta, delta, all the way down to Omicron variants. And what was going on there is that the variants in SARS-CoV-2 virus escaped the neutralizing antibody responses by accumulating mutations in the part of spike the spike was exploiting to infect the human cells. But largely, the T-cell response was unaffected. And why was unaffected? Because the T-cell response targeted the whole spike protein and, in fact, targeted the also all the other proteins in SARS-CoV-2. And so the mutations that were accumulated in the variants in particular parts of spike was a minor part of a T-cell response. And so what we saw is that in reality, the T-cell response can cross-react against different variants of the same virus, in this case, SARS-CoV-2. Furthermore, a number of different studies also studied the T-cell cross-reactivity beyond SARS-CoV-2, but cross-reactivity with other coronaviruses. And we showed very early on that there was this cross-reactive pre-existing memory T-cell response that we were able to map to peptides that were conserved between common cold coronaviruses and SARS-CoV-2. We also showed that people that had this cross-reactive pre-existing T-cell memory responded faster and better to vaccination. And a number of different studies pointed out that this cross-reactivity actually is biologically significant. Particularly, for example, I love this Sagar study in JCI, where they showed that people that had this pre-existing common cold cross-reactivity actually had better disease outcomes and had lower mortality. So having had a recent common cold infection before the pandemic started actually increased this cross-reactivity and had a real biological consequence. So here, the important point I want to underline is that so the T cells can not only cross-react amongst different variants of the same virus, but then cross-react against different viruses of the same family of viruses. So we were intrigued by these lessons we learned throughout the pandemic, and we thought that this may be a general concept where we can imagine to have a T-cell inducing component in a vaccine, and this could be used to generate a T-cell immunity with broadly targets conserved regions in the viral family. And this could have two effects. On one hand, oh, yeah, sure. Thank you. On one hand, you can focus the immune response on this cross-reactive region so that even if you don't know what is the virus that will be at risk of causing the next pandemic, if you're targeting a conserved region, then by definition, that should be found also in the virus of concern. And the other thing that you can do is you can broaden the specificity because, again, we were still thinking about that evidence that people that targeted more than one antigen had a better disease outcome. Last. And want to point out that this we envision is not should be done instead of inducing antibodies, but should be done synergistically. And because every time I talk about this, people say, oh, you just want to induce T cell because you're a T cell guy. No, I have nothing against neutralizing antibodies. I love them. But this is it's a different strategy that hopefully can be of significant. So what is the strategy? So first of all, we look at different viral families, as I was mentioning earlier. I mean, there's a bunch of different viral families that have been singled out by WHO and CEPI and other entities as potential concern. So, but within that one particular family, we want to select a prototype virus to anchor our investigations. And then, of course, you want to do a conservation analysis in the phylogenetic space. And this needs to be done properly in the sense you can't just say, download all the sequences from that viral family, because, for example, if you do that today for coronaviruses, you will essentially do a conservation analysis of Omicron, because people had sequenced so much Omicron compared to the other viruses. So you really need to think through, for that viral family, what is the phylogenetic space, so that you sample a representative sample of the viral phylogeny, one. Two, the other thing you want to do is you want to sample perhaps with a more carefully the viruses that are known to be able to infectious for humans, perhaps over sequencing viruses that are infectious for frogs. I mean, because ultimately you want to protect humans, not frogs. Then the next thing is you want to know what are the targets of a T cell response. If something is conserved but cannot be recognized by T cells, it's not of interest in this context. And you can either see if there is enough data already known, what is the target of T cell response, and if it's not known, you got to go and generate it. And then you want to put 2 and 2 together. And we specifically are interested in seeing if we can define conserved T cell epitope regions for the prototype virus we're studying. And then you will validate. So Alba Grifoni has recently published the studies where we validated this strategy in the context of common coronaviruses, both Sarbeco, beta coronavirus, and other coronaviruses. And so the data is published, and not going to go into much detail. One thing that I want to point out is that including this conserved T cell epitope regions beyond spike, and here we focused on beyond spike because what's the point of that? By now, everybody has been vaccinated with spike a million times. So what we want to do is something that goes beyond that. But you can see that if you add these non-spike conserved regions, you do the two things that I was interested in mentioning early, that you broaden the T cell response. And here is shown about a twofold gain in the breadth of the T cell response. And also importantly, you broaden the response across all different ethnicities. You see here, this is calculated data, but all the different main ethnicity worldwide, you really get a significant improvement in the breadth of response. Then the next thing want to do is to say, okay, we can combine all these different T cell epitope regions in a Frankenstein immunogen, where we take these different regions and we try to make them relatively big. We don't like to use just isolated epitope, make relatively big so that they have some degree of folding in smaller domain. And then we use AI and AlphaFold repetitively to just design things that have different stability, different stability features. And, oh, this got screwed up, didn't, okay. There was an animation, but very cool. We're testing them actually in mice. So one other good thing that comes out of making this big chunks of conserved T-cell epitope region is that they also have enough binding site that you can test them in mice. Because otherwise, if you just use isolated human epitopes, then they wouldn't work in mice. Okay, so that was coronaviruses. Another family of viruses that we are interested in are the arenaviruses. And you might, and arenaviruses have been singled out as a family with big potential pandemic potential. Perhaps the one that is more well known, it's Lassa. Lassa virus is actually a big problem in certain parts of Africa. Another member of the arenaviruses that is very well known because has been studied extensively in mice is LCMV. The arenaviruses really have two phylogenetic families. One is the New World, which are viruses that are found in Central and Southern America and have all these exotic names, Guanarito, Chapare, Junin, and so forth. And then there's Old World, which is Lassa and LCMV and other viruses, Mobala, and so forth. So we started out by looking at Lassa as a prototype and also the most immediate threat. Really, there is not a heck of a lot of data in humans for what is recognized by human T cells in Lassa. So we went ahead and generated data, and the way we generate the data is because we don't have access to large amount of blood from infected individuals, we do an in vitro simulation for 2 weeks to expand the cells that have a capacity of recognizing Lassa sequences, which is in a way it's doing a vaccination in the test tube. So you're essentially expanding naive T cells with pools of overlapping peptide by 15 months. If you do that with 15 months and IL-2, you get mostly CD4 T cells. And this is what is shown here, but the response is overwhelmingly CD4. We're also doing in parallel CD8 epitope identification. We're not going to talk about that today. So by doing that, we generated this first really high resolution map of Lassa human CD4 T cell response. I'm not going to go through the data in detail, but there are a lot of different epitopes that can be recognized by humans. If you are arenavirus immunologist, you know that one of the things that it's strange about arenaviruses is that the neutralizing antibodies develop very late. In fact, the neutralizing antibodies appear after the infection is cleared. So they have absolutely no role, which doesn't mean that you don't want to induce them. Yes, if you want to induce it to prevent the infection, but basically the clearing of infection is T-cell mediated. So it has been debated that maybe the reason why the neutralizing antibody are so late is that the glycoprotein, which is the surface protein of arenaviruses, doesn't have T-cell epitopes. And it is absolutely not true. A lot of perfectly good T-cell epitopes. Then you can also show, perhaps not surprising, that there's a strong correlation between the capacity of the peptides to bind HLA and their capacity of being recognized by human T cells. So then we did the phylogenetic conservation. Of course, we looked at many different isolates of Lassa, then non-Lassa old world arenaviruses and new world arenaviruses. And I'll spare you the detail of all the bioinformatic things of calculating sequence conservation throughout all the different things. So this is an example of the simplified data to just show you conceptually what we're talking about here. So here on the top is this particular region of Lassa. And you see this is 100% conserved in any different Lassa isolate you look at. And it's also very decently conserved in old world arenaviruses, in other old arenaviruses, and even in new world arenaviruses. There's some variation, but overall it's not bad. It's fairly conserved. And that is an example of a good thing. This is another example of a less good thing. So this particular region is very conserved in Lassa, but already when you go into old world, and when you go into new world, it's totally not conserved. So just to say that there is a breadth spectrum of conservation. So for further validation, what we did, we took a panel of different representative old world and new world arenaviruses. We expanded T cells specific for the different conserved T cell epitope regions, and then we asked whether the Lassa specific conserved T cell epitope region specific T cells would actually cross recognize the same region from all the different viruses. The other thing that you can do, because as I was saying, these chunks have usually enough binding sites for mouse MHC, you can actually make the Frankenstein immunogen and test it in mice. So I'm showing you here the cross-reactivity of the regions that have been selected for being conserved in Lassa, and they not surprisingly cross-react very well with other Lassa and with other old world arenaviruses, such as, for example, Mopeia, less well on LCMV. But importantly, they don't cross-react at all with the new world arenaviruses. So this tells us that old world arenaviruses are similar enough to each other that you can make something that is an immunogen that induces cross-reactive T cells. But when you go to the new world, they are already too different. Okay, so that's fine. Oh, okay, this also did not copy over well. What this was showing, if you would have seen the data, I'm sorry, but basically what you can do is do the same thing. So you make conserved Hunin regions as a prototype of a new world arenavirus, and those will generate T cells that cross-react in new world arenavirus, but do not cross-react with old world. So essentially the concept we're getting is that this will require a bivalent immunogen where you have one to cover old and one to cover new world. We perform additional validation. So this is mouse data where we made the Frankenstein immunogen, and we can show that, yes, we can induce T cells that are cross-reactive with other old world, but not new world arenaviruses. Okay. What this slide shows is further validation in humans. And so we have validation in humans in two different systems. So one is that a company called Emergent has generated an experimental Lassa vaccine, and we have samples, and this is a GPC delivered in a viral vector. And we obtained some samples from this vaccine trial, and we were able to show that the T cell induced in the vaccine recipient cross-reacted and cross-recognized the regions we have selected. Then the data you don't see, but would have been here, it's a really cool set of data where we have two, N of two, I understand, but we have two people that were infected with LCMV, and we can show that the people infected with LCMV cross-react the Lassa virus specific. And why is two? Because these are two lab accidents. So these are mouse immunologists that were working with LCMV and accidentally stuck a needle with LCMV in their finger. And they were generous enough to let them be bled by us. And we took their T cells to perform this experiment. And so it's two because you can't just immunize more people, infect people randomly. So these are accidents. So that is the bottom line of what we've done for Lassa and arenaviruses. And we can induce, identify this conserved T cell epitope region. And so we can show if this can be used to immunize in vitro and in vivo and get broad cross-reactivity between old and within new world arenaviruses, and but not across. And then this is, in a way, it's similar to what eventually we learned in coronaviruses, where you can cover cross-reactivity within sarbecoviruses and beta coronavirus and alpha coronavirus. They are a little bit different, so it's the same concept. So paramyxo. Okay, paramyxo, it's a different viral family that we're interested in. Here you see a list of some of the viruses that we've been targeting. So in the paramyxo is measles. So of course, there is a perfectly good vaccine for measles. But this is interesting because everybody is primed with measles. So you can study a memory T cell response. Then there is another family of paramyxo that are of interest and concern. And one is the prototype virus is that Nipah. Nipah, it's a very lethal, it does have outbreaks in Malaysia and in Asian regions. It's very concerning. And Hendra, it's another virus that is part of the same sub-branch of paramyxo, but is also of concern in terms of potential pandemic awareness. So they are similar, but not exactly the same. They have a overall similar viral structure and proteome. We did a series of experiments where we looked at the Nipah and measles epitope identification studies. They are almost the same, but not the same. Some people didn't have enough cells, so we have to drop them out and get a couple more donors. But essentially, by doing serology, you can see that the people that we studied, which again, these are local San Diego blood donors, they are negative for Nipah and Hendra and so forth, and they are positive, as they should, for mumps and measles and other paramyxo for which there is widespread exposure. So we did the same old thing. We did the screening. This is the N protein. was Nipah, and the Nipah epitopes are large number of epitopes, but there are a few that are really, really good. And we were able to map the restriction of class 2 for these various different Nipah epitopes. And same thing was done for measles, where we were able to map the response. So when you compare the immunogenicity for measles and Nipah, it's similar in terms of the pattern, but what you see is that the reactivity we see for measles is higher than what we see for Nipah, which makes sense because for measles, we are expanding a memory response that's already there because its people have been vaccinated, while for the Nipah, we're getting a primary response. And this is true also at the level of the specific regions of a proteome that are recognized. We do the same phylogenetic analysis of conservation for measles and Nipah epitope across all the different paramyxoviruses. This also is an example of overlaying the conservation with the T-cell immunogenicity. So here, as a heat map, is the conservation of all the different peptides that span the N protein. Obviously, I can show you almost all the different proteins, but I don't want to bore you to that. But point is that we see that there are certain regions within N which are much more conserved than other regions. Particularly this region here that is conserved not only across all Nipah, but also other non-Nipah and Nipah viruses, and also to a certain extent in other paramyxo of the essentially morbillivirus. And then you can actually see that correlates with immunogenicity for human CD4 T. So I think you can actually pick up some regions that are interesting, that are highly immunogenic and also highly conserved. And this is what you want to pursue. The other thing, this is a complicated slide, so I'll just take my word for it if you wish. If you look at the conservation, measles is really marginally correlating, measles immunogenicity marginally correlating with conservation, because the primary event shaping the T-cell response is the childhood vaccination. So obviously, when you vaccinate someone with a live attenuated measles, they don't know, they haven't seen other paramyxos, so they recognize what they recognize. While instead, if you take someone and you ask them what they want to recognize in Nipah sequences, they will preferentially recognize things that are conserved with measles because they have pre-existing memory against those. It's like common cold, corona, and SARS-CoV-2. So you're expanding a pre-existing cross-reactive memory. So we identified all these things, and the next thing you want to do is to do the same kind of validation we've done for SARS and coronaviruses, and for arenaviruses, and ask whether the conserved regions are cross-reactively recognized. And here you see the Nipah virus of different Nipah and Hendra, and the measles, and even the more distantly related paramyxoviruses. And when we did this series of experiments where we had Nipah expanded T-cell responses and asked whether they were able to cross-reactively recognize other paramyxoviruses, certainly there is a very, very high level of cross-reactivity between Hendra and Nipah and Nipah viruses in general, but also there is a fairly good cross-reactivity with still lower, but remarkable in other paramyxos such as measles, mumps, and so forth. And of course, this is translated in giving an opportunity to expand the targets of immune recognition and also conserving breadth of population coverage in different mechanisms. So overall conclusions are that this strategy of focusing on conserved T-cell epitope regions does give us an opportunity to focus the immune response on these regions that by definition allow good cross-reactivity in different viruses of a family of interest, and also to focus and broaden the response. We have at this point developed a general strategy for doing these experiments in any viral family of interest. And so as I showed you, arenaviruses, coronaviruses, paramyxoviruses, and I'm doing flaviviruses next, and polioviruses, and so forth. And just basically, it's like a well-oiled machine at this point. The thing that we constantly see is that within a viral family, it's probably generally true that you can cover family of viruses that are relatively related, but if you start to get too far, you will need multiple constructs, and also that there is a fundamental influence of the history of the exposure and or vaccination of the population you're studying, and that there is this constant phenomenon of exposure to whatever viruses depending on the family, and that kind of dictates and actually is in favor of expanding these conserved regions. And with that, I just want to acknowledge the many different families, many different individuals that contribute to the study. Professor Grifoni, an assistant professor at the La Jolla Institute, has really played a key role together with Ricardo da Silva Antunes, a senior scientist in my group, and many other people, in particular, the JCVI group doing all the conservation analysis, and Jimmy and his group doing the AI system design. And with that, I'd like to thank you for your patience. I'll take any questions.
Hello, thank you very much for your talk, and we apologize for the, I don't know, the technical problems with the presentation, but
Believe me, it's beautiful data, even if it's invisible.
So I got to see your data on the presented on the website. So there's time for questions.
Very much for the talk. So I actually have 3 questions, and I'll start maybe with the easiest, I think. So as a proof of principle, do you have data regarding whether those that had Nipah, whether they had the measles vaccine and they were more protected than maybe those that got Nipah and were not vaccinated?
So as far as we know, so these were all vaccinated with measles. So these are a local cohort from San Diego, La Jolla, where to the best of our knowledge, everybody got vaccinated in they still in San Diego is still popular to get vaccinated when you're birthed with measles. We're not in Texas.
But maybe you could get data from people who get Nipah, and as a proof of principle, check whether they were, depending on whether they were vaccinated or not against measles, whether they had the mild.
Yeah, it's so to get samples from Nipah infected people is not easy.
Know their status, whether they were vaccinated or not vaccinated.
So yes, so according to the places where Nipah outbreaks have occurred is Malaysia and Bangladesh. to the best of our knowledge, those countries has universal childhood vaccination for measles. So now the possibility exists that the people that are getting Nipah are the few exceptions that were not vaccinated. That doesn't seem to be the case in talking to people. Actually, the very interesting thing that the Nipah outbreak, the Nipah is transmitted by bats that feed on certain palm trees, and the outbreaks occur when they cook the sap of these palm trees, and they have communal thing, and then everybody but got that meal with the thing gets sick. I mean, could be that it's a family that chose. I mean, so anyway, short. I don't think so, but I don't really know. So
it wouldn't work then. I mean, then a universal vaccine or vaccines based on prototype viruses may not necessarily protect against those in the town, even though you see that you have cross-reactive.
right. So thanks for pointing that out. So our idea is that by specifically expanding those cross-reactive, you would get an additional benefit. If you just use the whole thing.
Go in then and you expand them.
Right.
There is any other question in the system? You're really welcome to, Alexis.
Thank you so much for the talk. I think it was nice you said to the families, because I'm sure the families have to tolerate the scientists, do so much time out of the homes, and that was a nice thing. So when you have these sort of cross-reactive T cells or antibodies that might recognize like emerging pathogens, do you think there's like a natural adaptive thing, or it's just chance?
A natural what?
Adaptive thing, or just it's like, you know, natural selection drives into an immune system where you have this cross-reactivity against like potentially emerging new viruses and bacteria, and or do you think it's just random?
So I think that it is a general phenomenon that you have some degree of cross-reactivity across or within a viral family. And I'm not sure if I'm answering your question correctly. And at the same time, also going back to the question that was asked earlier, I don't think that that is necessarily the best thing, in the sense if you want to prevent Nipah infection or SARS-CoV-2 infection, by all means, a SARS-CoV-2 or a Nipah specific vaccine is the best way to go. There is nothing better than a vaccine specific for the thing. The context I'm interested in here is, what if you don't know what is the next pandemic, right? So if you were in a pre-pandemic condition and you didn't know it was going to be SARS-CoV-2, but you had something that would be able to induce T-cell response that are directed against regions that are conserved between SARS-1, SARS-CoV-2, other sarbecoviruses, and so forth, that would be a first line of defense, kind of a less specific, but still broadly applicable to a family. And I have no idea if I answer your question.
You did, but that's fine. Do you believe it's random? Is it a random process? It's actually driven by some fitness feature for the immune system, having this potentially cross-reactive.
My guess is that it's random, in the sense that, so you can see the same. Now I get it. I think you can see the same thing in things that are not pathogenic. So, for example, maybe the clearest example you can see the same thing in allergens. So if you look at what people recognize in pollens, there is a tendency to recognize things that are conserved across different types of grass. And why is that? So I think so you get on one day you breed Timothy grass pollen and your T cells are expanded against all. But then the next day you breed ragweed pollen, and what you specifically expand is anything that is shared between those two types of pollens. Then next month you get still another pollen. So at the end of the day, as you go through cycles of exposure to different related things, you have a tendency to expand things that are cross-reactive or conserved. And so the idea here is that you do this even more on top of what happens naturally. You just basically push this. Did I answer the question now?
No, you did.
Thank you for your presentation, Professor. I wanted to ask you about your Frankenstein prototype of vaccine. I was wondering if you consider the use, let's say, 10 antigens with similar immunogenicity, is there a chance, or maybe you have seen in the lab, that there may be a bias immune response, either both humoral or cellular, against a particular antigen? Or if you have not seen it that in the lab, could it be possible?
Yes. So certainly that is another layer of complexity. And that is particularly relevant. So let's take antibody and T cells separate. That is particularly relevant again, in case where it's a re-exposure. And particularly for CD4 T cells, if you have different antigens and one is expressed at the very high level and one at a very low level, the CD4 T cells in particular will go preferentially for one is expressed at a high level of infection. And I did not show that, but that is certainly true for measles. So that the ones that are known to be expressed in higher have an advantage. For antibody response, then the bias is overwhelmingly on what is on the surface. And that's historically what is most immunogenic. And in fact, in our mind, we don't even go there. So and that's why I was showing a lot of times in the Frankenstein setting, the effect of adding that on top of the surface. So in our talking to different people, there is no way you will convince anybody of not doing a vaccination with a glycoprotein for Lassa, for example, because that is the main antigen on the surface. And so we're okay with that. So we're just saying, okay, fine, do the vaccine with GPC or do the vaccine for SARS based on spike. We're not trying to fight that. And say, but if you do a spike plus vaccine, so to speak, where you have spike, but you also have all these other conserved region that will also bring another dimension of T cell reactivity, you will do better. That's kind of the reason.
More questions?
Hi, thank you very much for your talk. Based on the data that you show us, specifically in the Paramyxo family, it seems that this strategy works better at the clade definition. I don't know if you have defined phylogenetic divergence threshold that limits this strategy?
So the threshold we use routinely, and we actually, that has by now is a fairly large data set, is that you want to have a degree of sequence conservation that is about 70%. And so as you drop below 70%, the T cell reactivity is no longer cross-reactive. That doesn't mean that if you're above, you will always be cross-reactive. It's a fairly good, so that's what we use operationally.
Okay, thank you very much.
Thank you very much for your talk. I have one question. When I saw your list of virus of concern and the virus that you use in your approach, many of them are monocatenary virus, not segmented. Only Lassa have 2 segments. And I want to ask you if this approach works well for other segmented virus as orthomyxovirus, for example, flu, because we are showing that in influenza, the change are also related with several segments, not only of HA or NA, also in the polymerases or in other conserved segments, but also have regions highly conserved across the genomes. So do you have any data related with this kind of virus?
Not really, but a little bit. I can elaborate a little bit on that. So conceptually, we could look into flu. We have not done flu with this approach per se. One of the reasons is that the universal flu vaccine is something that has been talked and worked on a lot for decades. And so I just wanted to do something that was less studied. That's one reason. The other thing is that flu is particularly difficult because it's like coronaviruses. So you have at this point such a high level of multiple exposure, re-exposures, vaccination that it's difficult. You have to do something really crazy good to go on top of what has been done. So that's but we have nothing against segmented viruses. Do you want to ask a difficult question now?
Is there any other questions? Back, students? González? No? Okay. Thank you. So, Alessandro, thank you so very much for your talk. And there is a coffee at the outside, and the people from the group of the facultad, the Grupo de Microbiología, we're going to take a picture. Let's go to the stage. Thank you very much.
Thank you. Those slides, I mean, I did.
¿Me escucha? Terminó el eventito. Exacto.